Welcome, everybody, to Science Says. Let me begin by pointing out the obvious. I am not Dr. Tom Lynch. Tom is the president and director of Fred Hutch. He holds the Ray Bacon-Dow Chair, and he ordinarily hosts this event. But Tom is reviewing another cancer center with the National Cancer Institute today, and he'll be back for next month's Science Says. So my name is Bruce Clurman. I'm executive vice president and deputy director at Fred Hutch. I hold the Rosbolt Reynolds Chair, and like many of the faculty here, I'm both a physician and a researcher. Just a minute about me. I came here in 1991 as an oncology fellow, so I've actually been at the Hutch for my entire career. In my lab, we work to discover basic mechanisms that allow cancer cells to grow and divide abnormally. We try to develop new approaches targeting these pathways to try and develop new cancer therapies. I'm also a clinician, and I specialize in medical oncology and bone marrow transplant and hematopoietic stem cell. And finally, as the center's deputy director, I work with Tom and our leadership and all of our faculty to develop and implement the Hutch's research strategy. One of our big areas of importance you'll be hearing about today has been finding ways to bring together researchers from throughout the center in different disciplines and bring them together to tackle some of the most important problems in cancer. So the Hutch is really an amazing place to do this kind of work. We have an exceptional breadth in the scope of our research, whether it's in cancer, whether it's in basic sciences, or it's really a defining feature of our center, and we do this because we don't know where the next breakthrough is coming from, but we do know it's going to come from outstanding science, and we need to be ready. You'll hear some of our work in COVID today. It's really an extraordinary example of how building a virology program allowed us to be in a position to make such important contributions to the pandemic. The scope of this research, I think, is also why we're the best funded cancer center in the country from the NIH. But I do want to point out that the NIH, despite the fact that it is why we're able to do what we do, doesn't really fund the kind of high risk, high reward research you're going to hear about today. And this is the kind of science that only happens because of generous donors, such as many of you here today. And we're super, super thankful for your support. So whether you've been to just one of these, if this is your first research forum or you've been to many of them, thank you for joining us. I hope you enjoy the hour. I also want to extend our gratitude to our partner for this event, Mercer Advisors, is a financial management company, and we're extremely grateful for their support. So we're going to talk about immunotherapy and COVID today over the next hour. We have 4 researchers we're going to talk with briefly, and we have lots of time to answer your questions as a panel afterwards. Let me just give a couple sentences about the immune system to get everybody on the same page. The immune system has obviously come to public awareness greatly over the course of the pandemic. There are 2 components we'll be discussing today, B cells and T cells. Both play critical roles in how our body responds to pathogens and to things like abnormal cancer cells. The B cells fight infections by making antibodies. These antibodies recognize proteins on the surface of cells that are being marked for destruction, whether they're due to pathogens or cancer. And the T cells are the other half of the immune system, which patrol the bodies looking for abnormal cells and either directly destroy these cells or instruct other components of the immune system to do the same. So our panel today is featuring researchers who are engineering T cells and B cells to fight cancer and infection. Some of the true pioneers in the field of immunotherapy, which is just a remarkable advance in the way that we're treating cancer. There's been amazing successes. You'll hear about one shortly, but we still have a lot to learn. We're trying hard to find new ways to make breakthroughs. One of the ways you'll hear about is our recent move into the historic steam plant next door to our main campus, where we're bringing together researchers from across the Hutch to accelerate immunotherapy breakthroughs. We're going to hear about immunotherapy, and then we're going to hear from Dr. Rachel Bender Ignacio, who is going to join us in our COVID corner to talk about COVID treatment trials and to answer your questions. So let me begin by introducing Dr. Stan Riddell. And so there he is. So Stan is a physician, scientist, and truly a world leader in the field of CAR T cells. He's really devoted his life to finding ways to re-engineer T cells to initially treat infections and now to treat cancer. I've had the pleasure of working with Stan for almost 30 years, and his work has really led to fundamental advances in T cell therapy, including the FDA approval of a Bristol Myers Squibb CAR T cell therapy that arose from his work. He works in the steam plant, and he holds the Burke-O'Reilly Family Chair in Immunotherapy. So thank you, Stan. So let me begin by asking you a question. I've been, over the course of our careers at the Hutch, it's been amazing to watch the evolution of cellular therapy from that of bone marrow transplant. I wonder if you could spend a few minutes telling us your impressions from your vantage point of how that's happened.
Yeah, thank you, Bruce, and good morning, everyone, and thank you for joining us. So it's interesting that Bruce brings up bone marrow transplant, because the reason that I came to the Hutch was a paper that was published from Fred Hutch in 1979 when I was still in medical school that described a phenomena called the graft versus leukemia effect. And that was that when you did an allogeneic bone marrow transplant, the donor's immune system was capable of eliminating the recipient's leukemia cells. And to me, I was interested in immunology, and to me that was just extraordinary. And it was at that time that I sort of set my sights on getting to Fred Hutch to train as a hem-onc fellow, and really to focus on trying to learn more about the graft versus leukemia effect, but also learn how we could use the immune system to treat cancer even without a transplant. So the very idea of immunotherapy actually grew out of transplant, and it was pioneered at Fred Hutch, through the work of Dr. Thomas and many of the scientists and physicians that worked with Dr. Thomas in those early years at the Hutch. So it was really instrumental, and I think has remained a guiding light, I think, and a guiding principle for all of us at Fred Hutch that do research.
Thanks. Yeah, it's been an amazing thing to watch, really. So the FDA just approved a Tcell therapy, Cell, that largely came out of your research. That must be an amazing experience for you. Can you tell us something about the early trials and when you recognized that this was really something extraordinary?
Yeah, well, first of all, I do want to emphasize that it was the research of many of us that had been working on developing CAR T cells. In particular, a close friend and collaborator of mine, Dr. Mike Jensen, who was at Seattle Children's. When Mike came to Seattle in 2010, we were both working on CAR T cells, and jointly, I think, really developed the approach that led to Lysocell. The first clinical trials were obviously started, before Lysocell, before Bristol Myers took over Juno, and was started even before Juno. And when we started these trials, I think we had, great hopes that this therapy would have activity. But just to remind everyone, when you do a first trial of a new therapy in patients, it's called a phase 1 trial. You typically take patients that are enrolled in the trial that have failed all other treatments, so they haven't responded to conventional therapy. So they're very courageous to go on these trials. And as a researcher, what you hope for is to see safety, that your therapeutic is safe and doesn't cause any harm. Of course, what you really hope for is that it'll have activity. And I can tell you, the first patient we treated, I'll never forget this as long as I live. And I got a phone call from Dr. Maloney, who was the clinician that was leading the trial. And he said, "You won't believe this. The patient's lymphoma is shrinking." He said, when I went in to see him in the hospital, he was feeling his neck because he had these tumor masses in his neck. And he said, "They feel like they're melting like ice cubes." And it was at that moment we just looked at each other and we just smiled. We knew that we had an effective therapy, and we just knew we had to optimize this, and it would ultimately benefit many patients. And that trial went on to enroll more than 200 patients with lymphoma, leukemia, with both acute and chronic leukemia, where we really defined, I think, the principles of using CAR T cells to treat patients with these advanced malignancies. And it is incredibly gratifying that it's now approved and being used to treat patients throughout the United States and eventually throughout the world.
It's just an amazing story and an evolving story. And so one of the ways it's evolving is by trying to take the work further. We're going to hear from Siobhani in a bit about taking it into solid tumors, but we talked briefly about the steam plant, and you and I had the opportunity to work together and thinking about what the steam plant might look like towards these goals. I wonder if you can tell us, what you're trying to do there, how it's working. We just opened it in the middle of a pandemic. So can you tell us about what the concepts are there?
Well, first of all, I think it's a real testament to the leadership of the Hutch to be able to actually open a new facility during a pandemic and actually have it really become fully operational. I'm actually not at the steam plant today. was there yesterday. It's extraordinary. I mean, first of all, the laboratory facilities are first class, but the idea behind the steam plant, the underlying concept, was actually to bring scientists together to work in an environment where they were not separated by walls, but could really closely collaborate. And in particular, what we wanted to do was to bring senior investigators with young investigators, and to bring many of the faculty that are working in immunology with our colleagues in computational biology. I spent yesterday afternoon meeting with a postdoctoral fellow of mine and with Rafael Gatardo, who leads our computational biology program, and one of his postdocs, and with Evan Newell, who's another scientist that does high-dimensional data analysis, to where we're going through data for a paper that we're getting ready to submit. This is exactly what we wanted the steam plant to be. It was that we were all there. We were all able to do it. We did some of us were in the room together. Some of us were over video conference, of course, because we couldn't all be in the room together because of the pandemic. But I think it really, for me, it was very fulfilling because it was the very idea, and we're doing it even in the presence of this pandemic. I think the steam plant is going to really be a catalyst for research and for what I call interdisciplinary research, which is to actually make use of all the expertise that the scientists at Fred Hutch have, to bring them together to really drive the next generation of therapies.
Thank you. It's really been amazing to see what's going on there. So Stan, will you stick around for our questions in a bit? We're going to talk to Shivani for a bit now.
Absolutely.
Thank you. So now I'd like to introduce the Shivani Srivastava. And Siobhani is one of our newest faculty members, but she's very familiar with the Hutch. In fact, she was a postdoctoral fellow with Stan in his lab before starting her own labs. She's also an immunologist. She works in preclinical models, helping to understand how T cells and engineered CAR T cells respond to cancer before, during, and after treatment. She's really focusing on a super important area, which is trying to expand the use of CAR T cells into solid tumors that represent such a huge cancer burden for people across the world. And so, welcome, Shivani, to join us today.
Thanks for having me.
Yeah. So let me ask you an easy question and a hard question. Okay. Here's the easy question. So you just became a faculty member here, and I know you could have gone anywhere that you wanted to go, but you chose to stay here at Fred Hutch. And I wonder if you can tell us from your perspective why you made that decision.
Yeah, so that's a good question. It's very kind of you to assume I could have gone anywhere. A lot of the work, as you described, that my research is really focused on is really trying to develop and improve adoptive T cell therapy for all human cancers. And a lot of that work is grounded in what we call basic immunology. So trying to use, preclinical models in the lab that really enable us to dissect out what are the mechanisms in the tumor that are really preventing engineered T cells from being effective. And then using that information to try to then rationally design ways to overcome those obstacles. But the other half of that work is tied really closely to clinical trials where, these therapies are being evaluated in patients, where we can see if, these ideas that we develop in mouse models are really going to have any real world impact in patients. And one of the things I think I realized is that there's really not a lot of other institutions and universities that really have the infrastructure to run these complex immunotherapy trials while also still excelling in basic research. Most places tend to specialize in one or the other. And at the Fred Hutch, with its history of adoptive T cell therapy that's been pioneered by people like Stan, as well as its history in basic science and, its ties to UW and the basic immunology community in Seattle, it's really one of the few places that's able to do both very successfully. So that made it a great fit for the kind of work that I want to do. And I feel very lucky to be able to build my lab here.
That's great. Thank you. We're very happy that you chose to stay here. So now comes the hard question.
Yeah.
CAR T cells have been remarkably effective in some blood cancers to date. But so much of the global cancer burden are solid tumors like lung cancer and breast cancer and pancreatic cancer, where we urgently need these therapies. And that's what you're working on. I wonder if you could tell us what some of the barriers that you're working on to try and make these therapies effective, and maybe where you see the field going in the next few years.
Yeah, so there's a number of challenges that really make solid tumors so much harder to treat than what we call blood cancers, right? Like liquid tumors. One of the first challenges is that you have to have an antigen, a molecule that's expressed on the tumor for the CAR to target. And typically, you want this to be highly expressed. You want it to be expressed on every tumor cell, and you don't want it expressed on any normal cells that you can't tolerate any collateral damage to. And that's something that's really difficult with solid tumors, because typically these are tumors that are developing from epithelial cells. So the cells, the kind of markers that they overexpress are also highly expressed in normal epithelia, which are the cells that line, all your vital organs, like the lung, for example, which you really cannot tolerate any toxicity to. So it's been a challenge finding kind of the right target antigen. But even in the situation where you have a perfect tumor-specific antigen that's, really uniformly expressed, there's still a number of challenges for T cells. With the example of leukemia and lymphoma, for example, those are tumors that typically reside in the blood, in the bone marrow, the lymph nodes. These are all sites where T cells normally traffic. So it's really easy for a T cell to find the tumor. With solid tumors, these are tumors that are growing in tissues, which are much more inaccessible to T cells, so places like the breast and the brain. So it's hard for the T cells to basically be able to exit the circulation and know that there's a tumor for them to go attack. And then even if they are able to infiltrate those tumors, solid tumors are able to develop this very suppressive microenvironment that's really evolved to try to shut down T cell function. And so you have this kind of, a sequential amount of challenges. So you have to find the right target antigen that's expressed on the tumor. You have to get the T cells into the tumor. You have to get them to be able to function long enough to be able to eliminate this bulky tumor. So really, it's going to require, I think, the right combination of treatments and engineering strategies to really be able to tackle all of these problems together.
And are you optimistic that we'll be making progress in the near future?
I think we're starting to see some signals with a combination of, we're really starting to understand more about T cell dysfunction, which has become one of the biggest hurdles in solid tumors. And I think people are developing really creative engineering strategies to try to preserve their function. And so I think we're starting to see signals where it's going to take the right synergistic combination of therapies. And I think it's just a matter of kind of figuring out what's the right cocktail.
Great. Well, thank you so much. And I hope you'll also be able to stay around for our panel discussion in a while. Great. Okay, we're going to bring on another one of our faculty members now. And this, I think, is going to be my biggest challenge of the day. So I'd like to introduce Jim Boonyaratanakornkit. So Jim is an infectious disease physician who cares for cancer patients who are coping with infections, particularly after stem cell transplants, where infections remain an enormous problem despite so much progress. He is a researcher in our vaccine and infectious disease division, and he's working to explore innovative new ways to prevent respiratory infections, which really are a remarkable, important, and dangerous problem for patients with compromised immune systems. He's working to understand why different people respond differently to the same infection. And of course, these types of differential responses we see every day in infections such as COVID-19, in addition to other pathogens. So Jim, thank you so much for joining us here today.
Thanks, Bruce.
So I have a first question for you, which is, you work on B cells. So we just heard about T cells that are being engineered to try and attack things like cancer. Can we re-engineer B cells in the same ways like we do T cells, or how else are you thinking of harnessing their power towards therapeutic goals?
Well, first, Bruce, let me just say thanks for tackling my last name. You pronounced it like a pro. Also, can I give a shout out to everyone who put together this Science Says series, and also to the person who came up with that awesome name, B plus T equals immunotherapy. I like that this person is very math-minded, but it's also really cool that they recognize the potential power of including B cells. So yes, we can definitely engineer B cells to harness their power. B cell immune therapy is a relatively newer field compared to the T cell side, but investigators at the Fred Hutch, like my mentor, Justin Taylor, are pioneers in this field. B cells are highly specialized factories capable of churning out huge amounts of proteins called antibodies. And these antibodies circulate throughout the body, and they can stick to other cells or viruses and inactivate them. So antibodies play a vital role in protecting us from infections and even cancer. And some of these B cell factories live and operate for a really long time, on the order of decades. So these two unique properties make B cells really intriguing targets for engineering to harness their power for medicine.
Great. Yeah, thank you. That is, obviously a hugely important and exciting area of immunotherapy. I wonder if you could talk about your own research a bit and the impact it can have for transplant patients, for the general population, and even for battling the COVID pandemic.
Yeah, I think this technology could be really important for our transplant patients who are extremely vulnerable to infections caused by their compromised immune state. So as a transplant infectious disease physician, I see these infections firsthand, and they are a menace because there's really very little we can do for a lot of these infections in this population. And when transplant patients are the most vulnerable, they don't respond well to vaccines. There are a lot of questions in the Q&A already about this. Now, what if we could give B cells engineered to produce protective antibodies at the same time patients are receiving their stem cells? So these B cells could basically act as a shield against infections. And I think this could have a significant impact on improving post-transplant outcomes and survival by reducing infections. You also asked about what this technology could do for the general population. And so there are some infections that, despite decades of effort, we still don't have a vaccine for. Or if we do have a vaccine, there's still a lot of room for improvement. So HIV and influenza are important examples. Another creative application is to engineer B cells to produce other essential proteins that are not antibodies. For example, this could be really helpful for patients born with metabolic diseases who are otherwise unable to produce enzymes on their own. So really, B cells are already pretty nifty cells, and we can engineer them as medicines for different diseases. And in terms of engineering B cells for COVID-19, my mentor, Justin Taylor, and I received really generous support from the Fred Hutch research donor community for a COVID-19 pilot 6 months ago to study B cells that bind to the virus that causes COVID-19. And one offshoot of this project is work being done by Matt Gray in our lab to engineer antibodies that can bind multiple respiratory viruses and tackle COVID-19 and its variants. So hopefully we can integrate this into our B cell engineering program for the benefit of our transplant patients who might not respond well to a vaccine.
Great. Thank you. Well, we're so thankful for all the work that you've been doing here for our patients and for the center. So as you mentioned, there's a number of questions that I'll be directing your way in a minute. So stay tuned. Okay. So we're gonna shift directions slightly, but not too far, and we're gonna move into our COVID corner. And I would like to introduce Dr. Rachel Bender Ignacio. And so Rachel is the medical director of our COVID-19 Clinical Research Center. So this is a center we opened in October. It's really a unique center, perhaps nationally or internationally, where it's focused on developing and hosting therapies to treat early stages of COVID-19 infection. She's an infectious disease physician at the University of Washington, where she's also the associate director of the UW AIDS Clinical Trials Unit. Before the pandemic, her research focused on the link between the immune system and HIV. This has really been an important theme here, which is that the virologists at the Hutch and at the UW who have focused on areas such as HIV were able to pivot so quickly to make such a huge impact on the COVID pandemic. And we're just so thankful for everything they've been able to contribute. Rachel's led inpatient and outpatient trials for COVID-19 therapies, which along with the vaccines, as we all read about in the paper, are part of very important therapies for ending the pandemic. She knows a ton about COVID-19, and I'm very happy she's here with us today. So Rachel, thank you for joining us.
Thank you so much for having me today, Bruce.
Yeah, absolutely. So I wonder if you could start by telling us, from what you've seen, what are the most promising treatments that we're seeing in the clinic for COVID?
Well, I think when we think about promising treatments, we think about several things. There's the scientific and the efficacy benefit, meaning they actually work, and then there's an entire additional layer of can we get them to people and how can we make them equitable and accessible for people. So, I think the most promising from an efficacy standpoint right now are monoclonal. There are two trials that we participated in here at the Hutch. Regeneron combination monoclonal antibody, as well as a Vir antibody. Those are both combination monoclonal antibodies, meaning they have two different targets against the receptor binding domain of the spike protein. So they target very similar places to where our vaccines do, but the idea is to give rapid passive immunity to people who are already infected prevent them from becoming sick and being hospitalized. So both of these studies have really shown that we can take people who are at high risk for hospitalization and prevent them from getting sick enough to need oxygen, developing pneumonia, and dying. And they're very important, but they're right now both of them are IV infusions, meaning we nurse safe place to have someone who's COVID positive administer these in time and space to watch them, and they're very expensive. So these are very, very important scientific contributions, but we're also working on other treatments that I think are exciting because of their accessibility and their applicability to broader population. The one is an antiviral Revere, which we participated in a phase 2 trial with the Ridgeback Therapeutics, a small startup or biotechnology company. And this drug is an antiviral that is effective against a variety of RNA viruses. It's an oral pill. It was very safe and very well tolerated. And I only have access to the published preliminary analysis that was presented at one of our scientific conferences earlier this year. But no one on this trial who had received the drug shedding virus after 5 days, shedding culturable virus, meaning after 5 days on this drug, we don't believe that someone can transmit it, and we do think that it could prevent people from getting sicker. And we're in the process of working on a bigger trial, part of a big multi-center trial across the United States to look at whether this drug reduces symptoms and keeps people out of the hospital. Again, very safe oral pill that was very well tolerated. So this is very exciting and has the possibility of potentially treating other RNA viruses like influenza. Lastly, we're working right now on a all-remote study called Stop COVID-2, which has the drug fluvoxamine in it. This is an FDA-approved drug that is part of the SSRI class, so commonly used to treat anxiety and OCD. And it has a really unique mechanism amongst that class that's different than some of the other drugs that actually interferes with some of the inflammatory pathways that COVID-2 turns on in a cell that lead to poor outcomes. And so there was a really exciting phase 2 study that showed that this kept people out of the hospital, and a really interesting real life study that many of you may have seen on 60 Minutes recently. And so we're taking part of this phase 3 trial to document whether we can keep people out of the hospital. This, again, very easily available, cheap, drug.
Great. Thank you. You know, you're breaking up a little bit on my computer, and so I'll repeat something that I can't hear. So far, I think it's been pretty clear. I know you've been we're going to talk in a minute about all the ethical and access issues around treatment that I know you've been so interested in, but I do want to ask you an unscheduled question, which is, do you think that we have in hand the tools that we need to successfully fight this pandemic globally, or do we need fundamental new ways to treat the virus? We have vaccines, we have the therapies you've talked about. You know, do you see the armamentarium we have currently as being sufficient to get our way through this?
What's currently approved, no. What's in process, yes. And I think the important part to remember is that science, the tools, but we also need, the political will and funding to make this possible. And I think what's really important with this is the idea that if we come up with a solution that works for the United States or for Europe, this is not necessarily a global solution. And as we're seeing now with what's happening in India is that, vaccine nationalism or therapeutic nationalism, meaning making our technology available in one place but not another, is really not going to lead to us.
Okay. You cut out there for a second, but can you repeat that last sentence of not going to lead to us?
I think that we really need to work against vaccine nationalism, against therapeutic nationalism, meaning guarding our discoveries, guarding patents close to our chest as a country or as a society, because we will not reach a solution to really any other pandemic. Think of our solutions and tech transfer, and the efforts of this really need to be done.
Good, thanks. So you've touched upon this briefly, some of the ethical issues involved globally in treating COVID. I wonder if you could tell us about some of the ethical issues that you face in your own treatment trials and how you're handling them.
Well, I think one of the most challenging things has been the speed with which therapeutics have been developed and approved, either conditionally through the emergency use authorization FDA has been using to expedite important treatments, or through full approval. And so what that means is that when we started work in the pandemic, first with inpatient studies, meaning people who are hospitalized, and now with people who are ambulatory or not sick enough to be hospitalized, we were testing all of our interventions against a placebo, because it's ethical to provide people a placebo when there's nothing that they have available that could treat them, that could prevent them from getting sick. What has been, I think, some of the most complicated work that we're doing is making sure that we are allowing people access to the standard of care, meaning allowing people to get treated with what will keep them safe, while also finding new scientific. Sometimes what the standard of care is, what is considered to be routine, can change on a day-to-day basis or week basis. So some of these trials have been sort of reconfigured mid-flight to make sure that we are not only able to keep our potential participants safe and make sure that they're accessing the currently available treatments to keep them safe, so figuring out ways to still look for a positive signal in a new treatment with everything else that's going on.
Well, thank you. Well, you're certainly having a huge impact on how we're fighting this pandemic locally, nationally, and globally. So thanks for everything that you're doing. So let's bring back everybody now, if we can, and answer some of the questions people have submitted. And let's please feel free to keep submitting questions. We have 25 minutes or so. So let me start with a question for Jim, and then have Rachel chime in. And this has to do with COVID vaccines, and I suspect COVID vaccines specifically for people who are immunosuppressed or have reduced B cell function from cancer therapy or from other reasons. So, Jim, what is the efficacy of COVID vaccines and other vaccines in immunosuppressed patients, and what can we do about that?
It's a really good question, and one that we're really trying to tackle here at the Fred Hutch. Immunocompromising conditions related to cancer span a really broad spectrum, so everyone is a little bit different. But in general, the thought is these vaccines are less efficacious. Now, I always argue something is better than nothing. And in some patients, they will mount a good response, and some patients will mount a weaker response. I can tell you about some of the research done by Josh Hill in our division on vaccines, influenza vaccines after CAR T cells. And so this is being studied very aggressively. And patients with weakened immune systems respond less well, but some do respond. So I would still encourage, I think our clinical infectious disease group is still encouraging our patients to get vaccines. Now, timing is important. So transplant patients, we generally will wait at least 3 months, although we're also looking at that too, like where did this 3 months come from? So this is really being actively looked at by our group and our division.
And Rachel, I wonder if you could tell us if there's any special role for the types of antibody or other therapies that you're studying for immunosuppressed patients, either to prevent or to treat COVID infections.
Yeah, I think that ultimately, as we are able to have more treatments and more accessible, that these monoclonal antibodies that we're currently mainly using for either people who've been recently exposed, so sort of a post-exposure prophylaxis, or for treatment, that we begin to use them in a little bit more of a long-term prophylaxis scenario. So for example, some of the monoclonals that are being developed right now, life of 3 months, for example. So we could expect a protection of quite a long time. And some of the new formulations are looking at subcutaneous or intramuscular injection, meaning just getting a shot as opposed to an IV infusion, which means that, I think these could be really great tools where we could find people specifically, like people who have impaired B cells, who we know might not make great immune responses, and give them a nice blanket of passive immunities. So these lab engineered antibodies kind of keep them protected for, 3 to 6 months at a time, or maybe longer. So I think that's coming down the pike. Right now, the urgency of using those in people who are actively sick is preventing that a little bit, but I'm hoping that we can move more into using them.
Great, thank you. So we're gonna shift to immunotherapy for a few minutes, and then we have some more COVID questions as well. So Stan, here's a more basic question that somebody has asked: is how does an immune cell manage to process all the information that it needs to recognize if a cell is normal or is deserving of a response?
It's a great question, and I was actually just typing an answer to that, but I'll answer it verbally here. So, one thing is, this has actually been something that's been studied in immunology, is how do we develop an immune system that is capable of doing this? And some of it has to do with how foreign antigens are presented to the immune system, how they're displayed. They're typically displayed on MHC molecules, and we develop, as our immune system develops, it's educated so that it recognizes what's self, and those T cells that could react with self antigens are eliminated, or they're regulated. So that's one mechanism. But when we engineer T cells with CARs, we can go beyond that, because now we can target any surface molecule. And so those molecules are often, as Shivani pointed out, often expressed both on the tumor cell and on some normal cells. And so one thing you hope for is there's a therapeutic window. But a more sophisticated approach, and one that we're taking with David Baker's lab at the University of Washington, is to actually instruct the T cells with what we call logic gating. So the T cell now just doesn't recognize one molecule, but it actually recognizes the constellation of molecules that are present on cancer cells that are not on normal cells. And in principle, and we've actually published on this now, this works actually quite well. And we're now working on translating it into first preclinical models and eventually to patients. So I think that's definitely an approach that I think holds promise. And it really involves pretty sophisticated protein engineering, as well as wiring of the cell to instruct it in ways that would preserve the normal cell and only target the tumor cell.
Yeah, it's amazing. It's amazing work. Siobhani, I have a couple related questions for you. A few people have asked about, the application of CAR T cells to broader therapies than the first patients that Stan described. And I wonder if you could talk about the applicability of CAR T cells currently to blood cancers in general. And then somebody asked about prostate cancer. So solid tumors, you've spoken some as well, but maybe you could address that.
Yeah, I think CAR T cells, because of, some of the things that we talked about of why blood cancers are sometimes a little bit more susceptible to T cell killing, because of where the tumor is localized and its accessibility to T cells. I think CARs will be great in many hematological malignancies, and so they're being developed now also for multiple myeloma with, I think, really promising clinical trial results. So I think CARs will be really effective in many different blood cancers. In the setting of prostate cancer, I think, prostate cancer has many of the same challenges that I kind of alluded to earlier. It's typically a very cold tumor, so what we call kind of an immune desert. So it looks, it doesn't have very many mutations in it. It looks very not foreign to the immune system. So the immune system typically has a difficult time detecting it as foreign. And so it has all these same challenges of T cells have a difficult time penetrating the tumor, finding the tumor, functioning within, et cetera. One of the advantages of prostate cancer is that for the most part, there have been a couple antigens that have been identified that are quite specific to the prostate. And so that's been kind of one hurdle that's, I think, has been able to kind of been overcome a little bit. So there are kind of clinical trials ongoing for prostate cancer using CAR T cells targeting some of these antigens that are quite prostate specific. But again, they're running into very of the similar kind of, difficulties we've seen with breast and lung cancer, which is just that the CAR T cells don't really penetrate very well and they don't function very well. So kind of the same strategies that we're using to try to understand what's shutting down their function, what's kind of the stuff that's inhibiting these T cells, I think those are going to become broadly applicable to many different solid tumors, prostate cancer included.
Great, thank you. I'm going to ask Stan one more before we go back to COVID for a few questions. So Stan, somebody asked a question which is highly related to some of your earlier work, which is, will immunotherapy cure Epstein-Barr virus or chronic Epstein-Barr virus infections? And you've certainly studied the use of T cells for related viral infections for much of your career. Can you comment on that?
Yeah, so we did study T cell therapy for a viral infection that, when I first came to the Hutch, was a real significant problem for bone marrow transplant patients. That being cytomegalovirus, which at that time would cause pneumonias that were fatal in as many as 15 to 20% of patients. So we developed a T cell therapy using T cells that we selected from the donor that were specific for that virus, and it actually worked quite well. And other groups then developed it to prevent Epstein-Barr virus infection after transplant as well. And indeed, that work is ongoing, and there are efforts to commercialize those therapies as off-the-shelf therapies for transplant patients. Now, chronic Epstein-Barr virus infection is a little bit different, and we really don't fully understand the pathogenesis and where the defects are in immunity. And Jim may want to comment on this as well, but so far we haven't really pursued T cell therapy for that, but I think it does, it is a constellation of syndromes. It needs to be looked at very carefully to understand where the defects are and where immunotherapy might be appropriate. It's not something my lab's working on right now, but I think it is certainly a significant problem, and one that I think immunotherapy could have a role for. That's
great. Thanks, Dan. So let's talk about COVID a bit more with some issues around immunosuppression, which is obviously a extremely important issue for our patients and for our physicians. And so, Jim, somebody's asking if they've had cancer surgery, and I'll extend that to other types of cancer treatments. Should people consider postponing vaccines, or where do you see vaccination fitting into active cancer treatment?
Thanks, Bruce. And like Stan, I was literally typing in the answer as you asked the question, about to hit enter. So I won't type any further. So yeah, it's a really great question. And it depends if the patient is receiving chemotherapy around the time of their surgery. So there's neoadjuvant chemotherapy, so chemotherapy given to shrink the tumor before surgery, or chemotherapy given after the surgery. So I think the surgery alone shouldn't have a significant impact on the timing of your vaccination. That person could get it before or after. It really depends on the chemotherapy they get. And what type of chemotherapy, if they are getting chemotherapy. The surgery alone, if it's a debulking process, I think shouldn't have a significant impact on the timing of the vaccination. And if you could just wait a week or so before or after, just to make sure you're not developing any side effects from the vaccine that might interfere with you going into the surgery. So some individuals post-vaccine develop fevers, chills. Everyone responds a little bit differently. When I got my vaccine, I had 24 hours of fatigue. And that was it. Some friends had some more side effects than that, and some people had no side effects. So I maybe give a window of about a week around the surgery, I think is a good idea. Rachel is also an expert in this area, so also value her wisdom as well if she has input in this area.
Sure. Rachel, would you like to comment as well?
No, I think, Jim, you covered it really well. I also work on our solid organ transplant at the university. One of the things that we're trying to do that is kind of part of our usual vaccination practice is encourage people who are about to become immunosuppressed to get the vaccines beforehand. So if we know somebody's going to get chemotherapy or a transplant and they're able to get vaccinated ahead of time, we think that gives their immune system the best chance of responding to the vaccine. And again, because none of these are live virus vaccines, it's safe to get the vaccines and then become immunosuppressed. And we we think that someone has the chance of making a better immune response to it before rather than after. That's not always possible based on vaccine availability, how urgent somebody's treatment needs.
So related to that, a few questions have to do with, do we or should patients be trying to understand the quality of their immune response by checking things like titers to vaccinations? And if they have not developed sufficient immune responses, would they recommend or be eligible for a third booster shot? How are you thinking about monitoring and dealing with inappropriate or unsuccessful immune responses?
I think really the first thing to make sure is that when people talk to their doctors, that this sort of standard COVID antibody tests that are approved actually look at nucleocapsid and they don't look at spike protein. And so even amongst, physician sort of chat pages, I've seen people say, hey, I have a patient who got vaccinated and they have no response. And we're finding that people have been testing for the nucleocapsid, which is not present in the vaccines at all, and is more of a marker of natural infection. So that's just the kind of important off the top comment. And the second one is there is a specific spike protein assay available that people can order, but we just don't have enough information, I think, right now to suggest that people have these measured and are using them, because studies are ongoing about boosters, both for new variants of concern and for people who might have a suboptimal. So I think the answer for right now is stay tuned. We may be suggesting those to be used clinically in discussion with people's, oncologists or primary providers in the future, but I just don't think there's enough data to be.
Yeah, I think that last point is really important. The field is evolving so quickly, and everybody is so different that patients really need to work closely with their care teams to understand how to best integrate these therapies with their own management of their own disease. So, Rachel, I do have another question for you. This was a specific one, but it is a really huge problem internationally. It has to do with vaccine hesitancy. So somebody mentioned that their 24-year-old grandson is reluctant to have a vaccine because they are concerned it might cause cancer in the future. And so, there's so many different reasons that people have hesitancy for these vaccines. I wonder if you could tell us how we reassure people around hesitancy that it's okay to be vaccinated and how we address this problem.
I think the most general comment is to look at scientific sources that you trust. I think the CDC, great information. The CoVPN, or the COVID Prevention Network, which is based here at the Hutch, also has really amazing resources in many languages geared specifically to speak to different needs. So I think, finding important, good scientific resources is really important, because there's just so much misinformation out there, and that helps. I think something that's really important to remember also is that any of these vaccines, even the sort of novel vaccines like the mRNA vaccine, the platform for these vaccines have been in development for over a decade. So even though the vaccine for COVID itself is brand new, it's been in development, and they slotted the genetic material for COVID in, or for SARS-CoV-2 in, and did the testing very quickly. But it's all been very rigorous, no corners cut. Essentially, we threw more money and more people at the problem to get it there faster, but there was nothing that was shortcut in any of the scientific.
Yeah, that's great. So let's move back to some immunotherapy as we start getting towards the end of the hour, and please feel free to continue to ask questions. So here's a question from somebody who works in the field in a different area, which is at Juno, who obviously was an important partner in BMS to developing CAR T-cell therapy. And so, Stan, I wonder if you could comment on the ways that industry and academia partner together to develop these incredibly complicated and expensive therapies. Yeah, you're on mute, actually. I knew I was going to get to say that sometime.
It's a great question. The relationship that we had with Juno. I mean, Juno was really founded by scientists out of Fred Hutch and Seattle Children's, as well as Memorial Sloan Kettering in New York. And the relationship that we had, where they supported the early trials, were really instrumental in actually making this an effective therapy. I think what I learned from that experience was that it is very possible for industry to collaborate highly effectively with academic centers. And indeed, Juno and BMS have continued to support research at the Hutch. One of the project I described that we were talking about at the steam plant yesterday was in part funded by some support that Juno committed to when we first developed the relationship, and is I think allowed us to be a little bit more adventuresome in the work that we do to try and identify either new targets or new understandings of how the immune system and the tumor is interacting. I mean, industry is really great at commercializing, these products once they are developed. But I think a lot of the deep and rich science that needs to be done to understand what the therapy should be, it can really be done in academia, and I think it's an important source of funding for research and an important interaction that we have.
Yeah, I mean, this is such an important and challenging area. I mean, a cancer center like the Hutch is very good at these exploratory proof of concept therapies, but we're obviously not in positions to develop, full therapies. On the other hand, we need to make sure we manage any, conflict of interest or issues around academia and industry. So it's a constant challenge to make sure that we do it correctly. And how do you think that has been going?
Yeah, I think it's been going very well. I mean, I think the Hutch is very transparent about the relationships that the investigators at the center have with industry. They're also, I think, put in, guidelines that I think are quite reasonable but strict, and so we know exactly what we can and can't do. I do want to mention one thing, because there was a question about the cost of CAR T-cell therapies, and I think this is one thing that we can't control with industry. When they commercialize these things, we can't control what ultimately they will charge for them. And I think that, for me, one of the disappointments was with CAR T-cells has been the cost, and I think it is going to limit the availability to many patients. And I think we can influence that by developing technologies that actually will reduce the costs, whether they be improvements in manufacturing or strategies for making off-the-shelf cells so that it doesn't cost as much to manufacture. And again, I think industry's been interested in partnering with us to try and, to do that, to make it essentially more cost-effective for patients.
Yeah, it's another big challenge, is access. So I'd like to ask Shivani one other question. So CAR T cells are one important mode of immunotherapy, but there's others. We haven't talked about many of the other types of immunotherapies and how you combine them with even more traditional or targeted therapies. And I wonder if you can talk about the potential for combination therapies when we're talking about treating cancer patients, whether they have normal immune systems or not.
Yeah, I think ultimately for solid tumors, combination therapies will be the way to go, because I think the chance that you can really engineer a CAR T cell to do everything that's required of a T cell is pretty unlikely. A lot of the work that we've been doing has been trying to understand, as I talked about, one of the difficulties with T cells is their ability to traffic into the tumor. And so one of the things we had looked at is, are there ways that we could actually induce inflammation within the tumor so that you can activate the signals that are normally used to recruit T cells, for example, during an infection? And so we had basically tested out this combination where you treat with a chemotherapy, you induce inflammation in the tumor. Now CAR T cells get into the tumor much better. They also can then respond to checkpoint inhibitors that can improve their, function. And so it's really kind of figuring out what are the kind of ideal combinations if we can kind of synergistically find the right combination of pathways to hit. And so I think those will be really important going forward to get any kind of signal of success in solid tumors.
Great. Thank you. So, Shivani, let me ask you a completely unrelated question as we think about what we're trying to do at the steam plant. And you've been living in the steam plant now. You've started a new lab there. What's it been like for you in terms of the potential and challenges and benefits? Yeah,
the steam plant is a beautiful building. It's a great time to be there in Seattle. We get a lot of water and sun views, but that's kind of a selfish reason to enjoy it. But one of the great things has been it's great to be around a lot of other labs that are also immunotherapy focused. I think part of what the pandemic has made us realize is how much of science and kind of those little sparks and ideas that kind of change the way you think about a problem happen from a casual conversation in the hallway and not from, a formal scheduled meeting. And so it's great to just kind of talk shop with the labs that are next door, that kind of thing. The other is like for our lab, we've been starting to dabble into these new technologies like single cell RNA sequencing, spatial RNA sequencing, where you really just get a ton of data that's just not possible for us to analyze on our own. And so having this, this great set of computational biologists at the STEAM plant has made it so easy to set up collaborations, and so I think that's really going to allow us to maximize the amount of information that we're able to extract from these data sets so that we can really try to understand, why are cars working in this context and not that context. So it's been great. Yeah.
Great. Thanks. So Jen, last minute question to you before I wrap up with a few things. I'll let you represent much of the faculty here at the Hutch. As a physician scientist, could you speak a little bit about how those two parts of your life inform one another and what being a physician has contributed to the research that you do?
Thanks, Bruce. Yeah, we have a great community of physician scientists here at the Fred Hutch, working with SCCA, University of Washington, Seattle Children's, and I'm so grateful to have the privilege to take care of patients. It keeps me busy. It also keeps me honest. I'm relatively new as one of the junior faculty, but I know really well the history of stem cell transplant that started here in Seattle that Dan Riddell mentioned earlier, and how far we've come as a community to improve outcomes for our patients through basic science, clinical research, and face-to-face patient care. It's like a whole team that puts this all together, kind of like the SCCA logo, that ring around the patient at the center. So it's an honor to walk alongside our patients and get to know them. And this grounds my research, also gives me a sense of urgency to get the research to the bedside as quick as possible. And of course, being a physician gives me insight into unmet medical needs and knowledge gaps that need to be addressed through research. And my patients are always teaching me through their lives, their stories, their journeys, and their questions. They drive me to search for answers, especially to the ones that we just don't have good answers yet. Even the Q&A, there's a bunch of questions that we're still actively looking at. And that's such a great thing to see. Keeps us busy for sure.
That was so well stated. I can't add much to it. So I want to thank everybody, the speakers and everybody who's joined us today. Particularly want to thank again Mercer Advisors who sponsored today's event, and all of you who have donated to our research at the Hutch. I hope you've heard a few vignettes of the kind of work that gets supported, the cutting edge, high impact work that only moves forward because of your help. And I want to remind everybody that Give Big, which is Seattle's 2-day online giving event, it actually starts 1 week from today on the 4th and the 5th of May. You can pledge your gift for that now on Fred Hutch's Give Big donation page. And when you give to the Hutch, as you've heard, you're fueling research, and we're fueling research that we're trying to bring to save lives as quickly as possible. So there's going to be no Science Says event in May, but Tom will be back in June to lead a conversation about diversity and inclusion and science at the Hutch. And so I hope you'll be able to join us then. In the meantime, please stay healthy and safe. If you haven't been vaccinated, please do it, especially one of our listeners' grandsons, please do it. And we hope to see everybody back in June. Thank you.